Thank you, Dr. Pandarangan, for the very nice. Thank you. It's indeed a great pleasure to attend this IAT conference. Initially, I have not attended many conferences. Let me be very frank about it. But subsequently, I felt that voice should be made in the conferences and educate people, bring people together. That was the main intention, particularly when we are sharing the word taxonomy among us. It's very essential that we all come together, exchange ideas, and encourage the youngsters to take our places as we progress. And it's not only, the youngsters also see that their youngsters are also progressing in the field that is required. And as I mentioned earlier in the remarks, that we need an army of taxonomists if we want to document our resources. Well, today's today's keynote address, I am not using any slides because I'm afraid that when I use slides, I may go astray and miss the real points that are required to be conveyed to the youngsters as well as those who are practicing both morphotaxonomy as well as molecular taxonomy. Of course, I don't want to overemphasize the need for plant taxonomy as it relates to diversity and relationships therein through identification, naming, classification, and in so doing, publishing a systematic floral enumeration of a given area. That's a simple task, but going to the field and executing this simple task is the most important aspect. India has the locational advantage as at the confluence of Indo-Malayan, these Eurasian and Afrotropical biogeographic realms, confers its floral exclusivity in addition to the considerable cohesions of the flora from the bordering countries. 4 of the 36 biodiversity hotspots which we share with the adjoining countries, and all of you are aware about that, and I need not have to go through that again and again, which are recognized by the Conservation International world over, spread across parts of India, and it is here that Indian flora is essentially concentrated and expectedly distinctive. The prospective managerial role of a taxonomist as a plant resource specialist is, in this context, is fairly rational. Morphotaxonomy is truly age-old, inherently appearing for being proximal to exploring nature and invariably linked to all other branches of biology. It was aptly recognized, as Madam Yadav said it, that mother science of biology. As it was the only active, please listen, it was the only active and practicing science of biology even before Linnaeus, the father of taxonomy, and sustained and survived till date. The subject is based on certain principles and established concepts and involves both primarily morphological observations and experimentation when pursuing inquiring into allied branches for harmonizing support. All of you are aware, palinotaxonomy, embryo taxonomy, anatomy in relation to taxonomy, all these aspects have been practiced in the past. The state of affairs concerning taxonomy was different 50 years ago, and many renowned teachers were primarily taxonomists and were part of the teaching faculty, inspiring students. Please carefully listen, particularly those who are teachers and those who want to choose the teaching profession and as a subject teacher of taxonomy. They were the ones elucidating students the nuances of divergence, similarities with adequate exposure to field studies to comprehend taxonomy in its integrity and perspective. We still remember, as biology students, running around teachers attempting to know the Latinized names of plants, charmed by characters and consistency, systematized variants in recognized plant groups. Such exposure fairly impelled students to embrace botany, taxonomy by passion and pursue the same as a profession. And let me tell you, I am one of them because that created passion. It is not just because of not only teachers who encouraged us, but in the villages where we were running around the bushes to know the names of the plants so that we can eat those fruits. Unfortunately, morphotaxonomy concerning angiosperms, I would say not only angiosperms, even other branches of botany, has taken retreat in several universities, colleges in the last 3 decades. Thoughtlessly, the teaching was neglected with curricular compositors, almost denying a deserved inclusion in various courses. The glorious past of botanical tours for plant collection, herbarium making, and their deposition in local herbaria are no more to be seen. And even if they are there, they are being totally neglected, except in a few colleges here and there. In most colleges and university departments, taxonomists as teachers are almost unseen across the country. In many universities, once the taxonomic teachers retires, and it was never replaced by a taxonomist, or they have never filled up positions in most of the state universities and colleges. And there is limited number of people coming to choose botany as their career subject. Monitoring committees in central funding agencies, please note, monitoring committees in central funding agencies are dominated by laboratory-oriented researchers and have no inkling of logistic requirements in the field works and often disregard or cut down genuine fund demands. I must tell you that there was a Northeast twinning project with the ICR system, and we were given quite a good amount. But imagine giving 45,000 rupees to travel from Bangalore to Northeast. That means Sikkim, Arunachal Pradesh, Assam, and Mizoram. These are the places I was to visit. 45,000 rupees. Can you imagine? And all the people sitting are mostly agriculturists and molecular biologists. They couldn't understand because they never traveled in Northeast or any place nearby. So this has led to silent death of taxonomists in nearly all universities and colleges. The knowledgeable and experienced taxonomy teachers faded away in the course of, thereby students lost the field linked interest. That's very important. But this going in the field and learning in the field and hands-on training, that is something which is unimaginable. Of course, when I say this, it is a general application. But there are still some teachers who really take to the heart the subject and teach them in such a way that students will not forget this experience. It is now well realized that taxonomy teaching, research, training have bearing on the documentation, monitoring, conservation of biological resources in the country. Partly, the policymakers are to be blamed for not creating opportunities for taxonomists despite their need in large numbers in bioresource documentation and sustainable utilization is very essential. If this condition persists, we have no one for the identification of our bioresources, which will place a risk on the sustenance of documented and undocumented bioresources. Much unfamiliarity prevailed both in students and even in teachers as far as the basics of botany and identities of popular plant species are concerned. Because after some time, if you continue to have only the modern subjects which have dominated the academic scenario, if that is there, we will not have somebody to identify even the roadside plants. That is what I'm trying to divert. Other supposed priorities of subjects grew at the cost of taxonomy. Teachers who have had taxonomy specialization, please again note, have also turned to molecular approaches for their survival, or biotechnological approaches or molecular approaches for their survival, recognition and career progression. Molecular biologists have no idea of procedures to be followed on how to identify a plant or animal species. All of them go by the familiarity with the material and its name like amateur persons. The unawareness is to such an extent that when a doctoral student in biotechnology was asked to get some legumes from the field by a member of the doctoral committee, the student asked the member whether the legumes are plants or animals. It really happened. And that student was a rank student. Means it is not student fault, it is the teacher's fault. You have to train them. Except those who have practiced classical taxonomy, then migrated to molecular taxonomy, there is a difference. Because they know how to identify the plant. This is the basic minimum which is required for any taxonomist. A Linnaean binomial is valued link about the species, including its morphology, nomenclature, taxonomic affinities, distribution, valued properties, and any other accumulated research, because it is tagged to that. Morphotaxonomists are not only authors of these binomials, but could ease their universal reference in communication. Their relevance as inventory builders is not only appreciated currently, and the doctorates in morphotaxonomy are not valued unless there is a slice of projecting molecular aspects in their thesis. A sad transformation in recent years is that molecular taxonomists are in preference to morphotaxonomists in all recruitments. Even in the academies, science academies, where our taxonomy people, the impact factor, they take into consideration, taxonomists are nowhere. Very, very limited, less than one. Whereas even a third-rate biotechnologist can have minimum 4 or 5 impact factor. I am not criticizing these people. They are a different class, and they have to practice as academicians, but they will not be able to help in documenting the resources which are disappearing every day. Multiple institutions were created to document and safeguard our floras, but not encourage taxonomy training to sustain them sensibly and professionally. Students turn to the other perspective subjects, failing appreciation and denial opportunities where they are due. Many foresters, this is another serious problem that we are facing. Many foresters, again, let me tell you, there are few exceptions, whatever, for everything I talk about. Many foresters are unfortunately non-biologists, owing to erroneous recruitment procedures. Their concerns and interventions are limited to by their unfamiliarity with the biota as a whole. They might turn conversant with few revenue-earning species of plants and flagship animals like the tiger and the elephant. So in the course of their service, their administrative control of the forest enables them to evaluate the appeals from the taxonomists for grant of or deny permissions for general or specific group explorations in the forest. Many of them are delayed. If you go to some of these states, in the southern states, we have experienced that they want you to deposit 30,000 rupees, 50,000 rupees for even granting permissions, as if we are going to deforest the whole area. While funding agents compel researchers to conduct explorations in scheduled time frames, because suppose by any chance, after passing all these hurdles, suppose you get a project, they want, you have to stick to time frames, but your permissions are not given in time frame. Most of the places, most of the times, what I have learned from many people is that permissions are granted towards the end of the project. So you have to refund the money because you are unable to conduct the field. The worthiness of taxonomy cannot be restored unless these issues are addressed and with genuine concern and all practicality. More importantly, waning of taxonomy happened not because of its style of inquiry, but because of the wrong appraisals of the few dispatched it all it on all fronts without knowing what is good and why it is indispensable. In particular, molecular systematic plants are classified based on evolutionary perspective, evolutionary relationships. In contrast to phonetics, these plants are grouped based on morphological similarities and dissimilarities or differences. The key constraints in phonetics is convergent evolution. It is feared that organisms that have independently evolved with similar physical traits but not closely related can be erroneously presented in one cluster. In molecular approaches, on the other hand, phylogenetic relationships are uncovered by recognizing monophyletic groups. So you are literally forcing everything into monophyletic groups. Molecular approaches and DNA taxonomy involve sophisticated machines, expensive chemicals in the form of reactants, primers, and enzymes, and statistical evaluation involving highly theorized mathematics and application of algorithms and computer programs. One can see many assumptions or limitations once one looks at the summary of procedures to generate species lineages or cladograms. The method goes like this. Leaf or other tissue samples are taken from chosen species and DNA, nuclear, chloroplast, mitochondrial, is extracted. They serve as the reference samples for the chosen species from which one or several genes or loci or regions are amplified. The chosen regions are replicated into thousands of copies in controlled condition using primers, DNA polymerases, and deoxynucleotides. This application happens in 3 consecutive stages, namely denaturation, annealing, and extension. A new duplicate double-stranded DNA is formed from each of the single strands, and the cycle is repeated multiple times to generate multiple copies. The PCR products, I am giving these details particularly those who are not practicing molecular taxonomy. The PCR products are subjected to Argos gel electrophoresis. The bands corresponding to different markers are cut and purified using purification kits. The ambiguous regions, poorly aligned positions in the sequence are removed by G blocks. DNA bases are read using appropriate technique. Sequences are aligned, pairwise sequence or multiple sequence alignment, using algorithms like Needleman-Woods, Smith-Waterman, or BLAST, that is basic alignment search tool. They are used directly to construct phylogenetic trees using various methods and as neighbor joining, maximum likelihood, Bayesian inference, and maximum parsimony. Instead of inferring trees directly from the sequences, genetic distances between sequences can be calculated and used to construct the phylogenetic trees. The tree represents the best estimate of evolutionary relationships among the taxa based on available data. Sequence comparisons are performed in several ways, and similarity matrix, distance matrix are calculated in sequence and comparisons to qualify the degree of similarity or dissimilarity between the sequences. These matrices are crucial to phylogenetics. Sometimes the data used to build cladograms, phylograms could be combination of both morphological and molecular sequences. In the morphological data, the presence or absence of shared derived characters is used to infer in evolutionary relationships. Morphological features can be binary or multi-state, like different shapes, sizes, colors, etc., and are used to construct matrices that represent the morphological variation across taxa. Morphological characters need to be coded for each taxon, and this involves scoring the presence or absence of specific morphological features or assigning numerical values to multi-state characters. Once the morphological characters are coded for each taxon, this data can be combined with molecular data to perform joint analysis that integrate both types of data. So cladograms are created, depict the relationships between different groups called clades. The nodes on the tree represents the common ancestors, projected descendants. The tops of the tree represents groups of descendant taxa, often species. Two descendant taxa that split from the same node are called sister groups. Outgroups stem from the base of the node. Typically, bootstrap or the other confidence estimate values are shown next to each other, above which represents a measure of support for the node. Bootstrap analysis is a resampling technique used to assess robustness of the inferred phylogenetic relationships. It involves sampling randomly from original dataset to generate multiple replicate datasets. Phylogenetic analysis is then performed on each replicate dataset to construct multiple trees. These trees represent variations in inferred relationships due to sampling error in the original dataset. These are indicative of validate or refute phylogenetic references. Again, there are some glitches in molecular analysis. As one reviews the whole procedure of molecular approach, there is more convolutedness in experimentation, circumambulation in data processing, in the evaluation of different taxa and their relationships. Researchers blindly go by machine dictates. Please mind, they blindly go by machine dictates, what is generated, and do not figure themselves the characterization and circumscription of the taxa which they investigate. Molecular inferences do not take into account the extinct taxa and predispose them as evolution in the evaluation. These inferences are prejudiced by the number of taxa taken into consideration in comparison with the actual numbers while generating cladograms. Lastly, there are innate weaknesses in the arbitrarily chosen regions of DNA, as they are not standardized or identical for species markers, specific markers in species delimitations in different groups. So each group you have to have a different sequence. Further, choosing the correct chloroplast DNA region is difficult because the lack of data on the speed of evolutionary rates among different non-coding cpDNA regions. In a recent evaluation of the phylogenetic utility of 21 non-coding chloroplast regions, it was observed that more variable regions have rarely been employed, and equally most widely used ones are among the least variable ones. It is truly unsound that no one, on one side, morpho-expressive characters in grouping the species, while on the other side, non-coding and non-expressive unmanifest genomic patches. Sorry to use the word patches because they are the loci which they normally are regions, gene regions which are used. So the sequence similarity and affinities. We illogically integrate these results and view them under one lens to build species lineages. In future, researchers are bound to arrive at different results based on selected markers for the entire genome they choose to use in the analysis, thereby reviving synonymy. This has serious effects on the nomenclature, creating genera, species, and even shifting or transferring species from one genus to another genus. The numerous options will be available in the future in the form of various markers. Their permutations and combinations, subjective decisions to pursue review taxonomic assessments. This might mess up the existing taxonomic literature, in particular nomenclature, concerning the generic and specific species use. Therefore, these procedures have no controls for genetic variability and differentiation in many species due to geographical isolation and environmental or ecological heterogeneity. Application of G-blocks or any similar tool to remove ambiguous regions in the DNA or protein sequences can indeed be considered a manipulation, but it is justified as it improves the data quality and enhances phylogenetic signals. The worrisome aspect is that of mathematics. So we all taxonomists have come to biology only because we fear the mathematics. The mathematics and algorithms applied are not usually within the comprehension of taxonomists. Phylogenetic trees are constructed from unrooted ones to rooted ones using bioinformatics software and algorithms. The trees are based on data furnished and that what is bestowed by the machine depending on the algorithms applied. This is a very important aspect that we should really think about. They are considered close to evolution, and this stated with the certain degree of confidence. Evolutionary studies and phylogenetic inferences may be usually in different contexts, but name changes proposed, particularly in generic and specific or infraspecific levels, are disturbing. We are least convinced that the binomials should imbibe such evolutionary history or lineages and bring in such changes that turn out to be destroy long existing and established names. People may wonder whether classical taxonomy has a suicide relationship with the molecular taxonomy. In the name of monophily, both mergers and splitters were done by different genera destroying the established field of taxonomy. An illustrative example, this is another two examples only, but there are thousands of examples. I am giving only just two examples. These examples relates to nuclear and chloroplast DNA study in combination with morpho and paleontological characters to resolve phylogeny in the genus Desmodium in legumes. Because being a legume specialist, so I have taken these one or two examples. As many as 34 split genera are made from Desmodium. I don't know how many of you are aware that there is no Desmodium in India. Now, all of them they have gone into 34 different genera. And these 34 genera actually represented previously as subgenera or subspecies or species or even sections. So this is the situation that we are facing. And changing nomenclature so frequently is a real headache based on a simple molecule. Out of 350 species under Desmodium, 179 are recognized as Desmodium sensu stricto, and all of them are confined to tropical South America. What we have, only 4 weeds which are introduced in our country, and particularly mainly in the rubber plantations and coffee plantations in southern region. All those 4 species are nothing but Intortum, Desmodium Intortum, Desmodium scarpiurus, which has now become almost a weed at the national level. Desmodium tartuosum and Desmodium uncinatum, which are used as green manure crops. So one can envision the weirdness this work has brought into literature and number of names or name changes that have bloomed and withered. Either they have bloomed or they are withered. A similar work relates to phylogenetic reconstruction by Bruins and his group, in which 165 species of Brachystelmas and 194 species of Tepiliades were included under Ceropegia. Just imagine, we have Iraceum, for example, in Asteraceae, which has about 5,000 species. I don't know how many of you are aware that one composite genus has more than 5,000 species all over the world. That's the largest known genus. Because prior to that, even I was knowing only Astragalus, a legume genus, which is the, I thought it is the largest genus, which has about 3,400 species. But how to handle these species? These are all known species, documented species we are handling. So how do you identify? Now, let me tell you, the Astragalus genus, there are about 128 sections which are made, and the maximum diversity is in West Asia, and the second maximum diversity is in North America, and third in China. And we too have, it is one of the largest legume genus in India. There are a large number of problems in this. To even identification is a very tough job. And it is here we want that molecular taxonomy people should come and help us to resolve these complexes. It is there that the usefulness of molecular taxonomy can be heard. There might be typification and name issues with changed perceptions in the delimitation of different taxonomic groups. Further, there would not be any finality to such enquiries, and there can be yet again discordance in the judged phylogenies depending on the chosen region of DNA for analysis. This will create serious problems in the nomenclature stability of the plants. All of you are aware that nomenclature stability we have reached almost more than 300 years it has taken from lineage to date. And still lot of amendments are going on, and we are refining but we feel comfortable that at the end of the day we are close to almost final the ICM that we have at present. Good taxonomic revisions were supposed to critically examine the types of correct names and synonyms in literature as a standard taxonomic practice to make appropriate and right decisions. The molecular taxonomists deposit their voucher specimens in different herbaria alongside molecular codes in specified gene banks. This has implications for reviewing procedures or nomenclature decisions and revisions. This has implications in reviewing procedure. What would be the guidelines for future reviewers in accepting conventional standard type material and the vouchered specimens referred by the molecular reviewers? While erecting new genera and species, if there are multiple decisions based on different markers, different vouchered specimens, what should be the approach of taxonomic revisions? Whether the revision is customary will persist in future. What will be the situation when the whole genome is sequenced? Reportedly in China, they are supposed to map the entire genome of every species that they have. What will happen to the molecular taxonomy or selected gene loci which we have practicing at present? For all species against the selected loci or patches of DNA at present. There are some beneficial influences on traditional taxonomy when the data derived from other branches of biology such as cytology, palynology, anatomy, embryology, and you name a number of other went into it, but these branches could not survive on their own because they were always in relation with the morphotaxonomy. But you see today how many are practicing the psychology or taxonomy, anatomy and palynology. They are already disappearing completely. There are some, the molecular taxonomists who are bereft of taxonomy, basic taxonomy background, cannot handle explorations, enumerations and flora independently. Field exploration should be part of the sustained taxonomy work. The core taxonomy institutions should continue to have their taxonomy experts as before. Our recruitments should focus on morphotaxonomists with some element of experience in molecular taxonomy and not vice versa. Our updating with modern tools, including molecular approaches, should be situation specific as needs arise. and more precisely when taxonomic issues identified as complexes based on morphotaxonomy are rightly recognized for molecular study for possible better resolution. We are naming obvious perspectives and visible or microscopically observable forms are not cryptic or hidden or invisible or formless subjects. In classical taxonomy, what is practical will survive, others will fade away. Will the molecular taxonomy go into oblivion in the same way for the sole reason of impracticality of its application and independent of morphotaxonomy? So really, I feel, because when I was the director and representing on the steering committee at the DBT, Professor A.K. Sharma, all of you are aware, was the chairperson, and we started a program on molecular taxonomy, and sufficient funds were given. As I mentioned, reactants and other chemicals, these things are very expensive ones, whereas taxonomist needs just a gunny bag. He has to just pluck the best material and bring them. So we have given sufficient funds, and there was never a marriage between a morphotaxonomist and a molecular taxonomist. The program was abandoned. Let me tell you, the program was abandoned because the molecular taxonomist is singing his own song, the morphotaxonomist is singing her own song. There was no marriage between the two. So this is where we require. We have to work very closely, understand things. Either molecular taxonomists should know basic taxonomy, or morphotaxonomists should know molecular taxonomy. Then, yes, there is a possibility, and it is not for general application, and it is mainly for complexes where we are not able to resolve based on the molecular morphological theories. So this is what I want to try. It was not my presentation, and this, let me tell you, the entire presentation is coming as a paper in current science. Only yesterday I got the approval or acceptance letter. And this, my colleague Dr. Venu and myself, we debated with each other for almost 7-8 months, not face to face, sitting in chamber together, but he was in Hyderabad and I was in Bangalore, and we over phone for 2 hours or 3 hours at a time we were talking. And that's how and molecular aspects, let me tell you, I am not at all good at. He was able to comprehend by discussion with Ritesh Chaudhary and Dr. Epake Pandey and several other people. So that is what I want to drive at today, and how the molecular inclusions have happened in morphotaxonomy, which many people are practicing morphotaxonomy. I don't say that molecular taxonomy should not be practiced. In practice, you have to, but identify areas where morphotaxonomists could not resolve. It should not be that wherever I have doubt, I also have doubt in the same place. That should not be the attitude. Thank you very much.